Okay, this PowerPoint is kind of the last chunk of the heredity unit, and it's really going to focus on sex-linked disorders and other things that don't really follow Mendel's rules. So first, the chromosomal theory of inheritance is kind of just a review or refresher, is that genes live in specific locations on chromosomes, and we had talked about that before. And you get half from your mom and half from your dad. And there are different versions of those genes called alleles. And the idea here is that chromosomes segregate and assort independently. So when they are lining up and they separate and blah, 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 all of them do it independently. So you end up with unique combinations of things. In the picture, you can see that one particular gene has been stained. So you can kind of see those would be the homologous chromosomes that a person has for a particular gene. So once upon a time, someone named Thomas Hunt Morgan really loved studying fruit flies. And so fruit flies are like one of the hallmark genetic organisms that get studied, kind of like Mendel and his pea plants, but fruit flies are, I guess, closer to humans, right? So we use them a lot. I have horrible flashbacks from my college years of having to work with fruit flies in genetics class. But the idea is they only have 4 pairs of chromosomes, so it's easy to see, and they're really fast breeding. A lot of things that are associated with fruit flies, or Drosophila melanogaster, is their scientific name, Drosophila is what they oftentimes get called, is that some of their genes are sex-linked, which means they are located on an X chromosome or a Y chromosome. When we start to talk about humans, all of the sex-linked genes and traits that we usually talk about are on the X, because the X chromosome is much larger than the Y. Remember, the Y is really tiny and really only has a few handful of genes. But some examples of fly sex-linked genes include eye color. So wild type. The word or phrase wild type really means that's what is out there in the world. That's what's considered normal in a wild fly. So it's kind of the comparison. So whenever you see wild type, that's just whatever the normal is. So red eyes is the wild type for a fruit fly. White eyes would be considered the mutant version. And this specific gene is carried on a specific chromosome. It's also important to note that sex determination is a little bit different in certain organisms. So for us, as humans, we have the XY system. 2 X's means you're biologically a girl. An X and a Y means that you are biologically a boy. And this results from egg and sperm coming together. And depending on what sperm you get one or the other. Things like grasshoppers actually have an X0 system. So there is no Y. It's just if you have 2 X's, then great, you're a female. If you only have 1 X, then you're a male. Things like roosters have a ZW system. So if you get a Z and a W, then you're a female. Keep in mind this little symbol right here that looks like a little person. That means female. And then male has the little projecting arrow. You can remember it however, but that's what those symbols designate. ZZ for a rooster would be then you are a male. And bees are really interesting because if you are diploid, meaning you have double the genetic material, then you are a female. And if you have half the genetic material, then you are a male. Okay, so sex-linked genes. Here's a picture of an X chromosome and a picture of a Y chromosome. Again, you can see that it's pretty tiny. Most of this you know already, but the idea here is that fathers determine sex with their sperm. So they are going to either pass on an X or a Y. Most of our sex-linked disorders or genes or traits are located on the X chromosome. So fathers give their daughters an X. So therefore, they are passing all of those genes to their daughters, but they don't give them to their sons. Males express recessive traits a lot because they get a Y chromosome from their dad. So really, they are solely relying on the X genetic material from their mother. So that's why it's a lot easier for males to express a recessive trait that is located on the X chromosome, which is why males, as we'll find out, are more prone to sex-linked disorders. Females oftentimes can be a carrier because they could have gotten a bad X from one parent, but then they still have that second X that they can then mask whatever the disorder might be or whatever the trait might be. Okay, so here's an example of what that might look like. And whenever we're talking about sex-linked traits, we always write it in this fashion. So the female is XX, and the trait itself is written as a superscript. So big N, big N. Or if you're a male, you automatically get XY, and we write down the trait as a superscript as well. Only on the X, though, for humans. As I mentioned, there's really nothing on the Y, so it just gets a Y. So you create Punnett squares just as you normally would. In this case, we're shading because the little n might be a specific disorder. So this male here is carrying the disorder because they only have one X and it's recessive, and they don't have another X to mask it. Mom is totally fine. So then you have your combinations. Dad can either give the X or the Y. Mom can only give X's. And then you end up with this combination of children. The light pink is showing you a carrier. So these females are carrying whatever recessive trait is being talked about, but they also have an X that's masking it. So they might not show whatever disease or trait it is. Now, if mom was a carrier and dad was totally fine, that's a different story, because then mom can either give the good X, so to speak, or dominant, or recessive X. And dad can either give the dominant X or a Y. So there is an instance in that combination where you would end up with a male that has the recessive trait. And then if dad has the recessive trait and mom is a carrier, you end up with even more situations here of those that have whatever the trait is. In this case, now you start to get females that have it. So things like this include colorblindness, hemophilia, Duchenne muscular dystrophy. Again, it's a lot easier for a male to get a sex-linked disorder or display a sex-linked trait. Male pattern baldness is another common one because it's on the X chromosome and males only have one. Okay, another concept to talk about here is something called X-Inactivation. And it's kind of interesting because females are XX and males are XY, but having two X's is a lot of genetic material. So what ends up happening is that the cells actually inactivate one. So humans can live without an X chromosome. If you have an X and nothing else, it's called Turner syndrome, and you're mostly fine. But females who have XX, they actually inactivate an X chromosome. Again, this is just to regulate gene dosage. Having two full X's is just too much genetic material. Similar to, Down syndrome, having a whole extra chromosome is just too much genetic material. So it's a dosage problem. So we inactivate one, and it becomes what is called a Barr body. So early in the embryo, it's kind of interesting. This is a cat example, and the allele for fur color is on the X. So a cat could inherit, one version that has an allele for orange and one that has an allele for black. So as the cells are dividing, they randomly inactivate one of the X's. So in doing that, some of the cells are essentially inactivating the orange fur chromosome, and some of the cells are inactivating the black fur chromosome. And that is how we actually end up with calico cats or tortoise shell. because of the random inactivation of X creating Barr bodies. Some of those are going to express black and some of them are going to express orange. And it's important to note that really only females do this because they have two X's. The males, they only have one X, so they would just be a singular color. So fun fact, any cat that is calico or tortoiseshell must be a female. Okay, so in human development, the Y chromosome does have an important function. It makes a boy a boy. So it is required for the development of testes. So if you have a Y chromosome, it will create the development of testes. Embryo gonads are indifferent at 2 months. So there is literally zero difference between a male and a female up to that point. Then there is one particular gene called the SARI gene. I don't know, a female must have discovered it, I guess. The SARI gene is what sex determines a male. And this is like the one key instance where it is considered Y-linked because it's only found on the Y chromosome. And it codes for a protein that can regulate other things. Basically, it turns on all of the necessary pieces to make a boy a boy. Circling back to overall genetic recombination, sexual reproduction and the way that we reproduce yields a lot of opportunity for genetic recombination, which is kind of evolutionarily advantageous. We get offspring with new combos of genes from the parent. If the offspring looks identical to the parent, it's called a parental type. And if it looks different than the parent, it is called a recombinant. So, for example, if I am crossing a yellow round parent and a green wrinkled parent, any offspring that are yellow round or green wrinkled would be considered parental type. And then when you get new combos that don't really look anything like the parents, those are called recombinants. We're now going to bring up the idea of something called linked genes. So if they do not follow Mendel's law of independent assortment, then it is something called linked genes, where two genes seem to travel together because the ratios that you would get are not what you would normally expect from a random dihybrid cross. Similar to on our previous experiment, like round and yellow and green and wrinkled make that 9-3-3-1 combination because of random assortment. If you get something different than that, then it's probably because the genes are linked together, and I'll explain that in a minute. So here we're going to be looking at fruit flies, and a wild type or normal version would have a gray body and normal wings. So for wild type, oftentimes you'll see little pluses, which means they have whatever the trait is. Now, if we have this double mutant over here, it's black instead of gray, and it has vestigial wings, which means that they're tiny little shriveled up wings compared to normal, and they don't have any pluses. So what you do in this instance is we cross them, and we get all of these flies here. And it's heterozygous because it got all of the pluses from mom and the not pluses from dad, we'll say. So then you take that times the double mutant, and you get this combination of things. The sperm or the dad can only give little b, little vg without any pluses, and mom can give any of these combination of things. So if you're predicting what would have happened if genes are on different chromosomes and everything is going all random like it should, you should get an even distribution of all four of those categories. However, that is not what happens. More often than not, you'd end up with the parental types. So you end up with a lot of wild type gray normal ones, and you ended up with a lot of black vestigial ones. And it's not impossible, but you get a couple gray vestigial and black normal, but not as much as you normally would expect. So kind of cut off here. The actual numbers are kind of 900 and somethings to about 200, which is not what you normally would have expected. So this brings about the idea of linked genes. Genes that are located on the same chromosome, which are really close together, tend to be inherited together, just because of their location. So because they're on the same chromosome really close together, then they tend to move around together. You can see here, if I had the female that was heterozygous, then oftentimes most of the offspring will have gotten one, either both pluses or both without, and then the combination from dad. That's just kind of what most offspring end up being. And you didn't end up with 0 other combinations because of that idea of crossing over. So during Meiosis I, if we remember, when tetrads are formed, so homologous chromosomes line up together, so you have like mom's version and dad's version with its copy, crossing over can occur. So if these are the genes that we're looking at, it is possible that this chunk swapped. So now you have a new and different combination, these recombinants, that you didn't have before. So it is possible. But the principle here is that the farther apart 2 genes are, then there's a higher likelihood of crossing over, meaning you'll get more recombinants. But if you are super close together, then the likelihood of crossing over occurring that would separate 2 really close genes is very rare. So that recombination frequency would go down. Long story short, the closer 2 genes are, the less likely crossing over is going to take place and the lower recombination frequency you're going to have. The farther apart 2 genes are, well, then the higher crossing over and the higher recombination frequency. If they're far enough apart, then they're really just at that point independently assorting. Okay, so there's a way to put a number on this. So these are pulling from the same numbers a couple of slides ago. So we have the normal versions or the parental types, and then we have the few that occurred, that must have occurred, I guess, via crossing over. So to calculate this, or the recombination frequency, you look at how many recombinants occurred out of the total number of offspring. So in this case, there were 391 recombinant options by adding the 2 types together, out of a total of 2,300 offspring, which yields 17%. So in this case, the recombination frequency is 17%. A higher percentage would mean that the genes are farther apart. A lower percentage would mean that the genes are closer apart, because it's harder to get a recombinant that way. So we can use this fact to generate something called a linkage map. By observing the number of recombinants that occur by crossing different traits together, you can really get a picture or start to organize where genes must be on a chromosome. So the idea here is that a 1% recombination frequency translates to kind of like 1 map unit, or 1 step, I guess, on a chromosome. And it helps give a relative distance. If you have anything 50% recombination or higher, usually 50% is the high, then that means the genes are either really far apart or they're on different chromosomes. But anything less than that, you can try to organize them by their recombination frequency. So, for example, previously we had color in wings that were 17%. recombination frequency. So they would be kind of 17 map units apart. If I then added in this other gene called CN, and I found out that between color and CN that it's 9%, and that between wings and CN it's 9.5, now I can actually get a picture of where that might be, and you can start to organize them. Okay, so the last couple slides have a couple other vocabulary words that we'll talk about later also in Unit 6. There's something called genomic imprinting. And this is where there is a phenotypic effect of a gene depending on if it came from mom or dad, male or female parent. There's also this idea of methylation, silencing genes by adding methyl groups. So certain genes, for example, if the mutant allele, we'll say, is inherited from mom, you have a normal-sized mouse because the version of the normal one is expressed and the mutant one is not. But if you inherit the mutant version from dad, you get a dwarf mouse because the normal one is not expressed and the mutant one is expressed. So this is a weird instance of, depending on where you get the mutant version from, mom or dad, and the inactivation of certain genes, you have different outcomes. It's called genomic imprinting. There's also something called non-nuclear DNA. And you kind of know about this already, that there are some genes or genetic material that lives outside of the nucleus. It's not in the chromosomes. So this is found in the mitochondria, remember. It has its own DNA. Chloroplasts do. Thinking back to the endosymbiotic theory, they both have their own genetic material. Plastids or plasmids are other examples, which are small circular pieces of DNA that things can have, like bacteria. And as I mentioned before, when we talked about mitochondrial DNA, you get all of your mitochondrial DNA from your mother because it lives in the egg. And depending on a combination of those things, you can then express different traits. Here's an example of a striped or spotted plant results in a mutation that happened in a pigment gene in a plastid. So these are oftentimes maternally inherited, but if there's a mutation in those particular genes, then you can end up with differences as well. Okay. At the end here, it just talks about genetic testing, just because there's a lot of things that you could do to test future children for different types of disorders. Here's just kind of a list. Two common ones include amniocentesis and chorionic villus sampling. So this is just kind of extracting some fetal tissue or fluid from around the fetus that has genetic material from the fetus in it to do something like a karyotype, which we learned about previously at the end, that displays all of the chromosomes. So you can get an idea of what potential chromosomal disorders a child might have. And how do you arrive at a chromosomal disorder? This is an important thing called nondisjunction. And in nondisjunction, what that means is the chromosomes fail to separate correctly. So this can happen in Meiosis I, can also happen in Meiosis II. So in the left here, what could happen is when the tetrads line up down the middle in metaphase I, the homologous chromosomes could fail to separate. So what that means is that all of these end up going to one cell, and that you'll get half as much in the other cell. And then they'll go through meiosis II normally, and you're going to end up with some gametes that have extra genetic material and some gametes that are missing genetic material. The process could also happen in meiosis II, and that's where the sister chromatids are going to fail to separate. So in this case, you're going to end up with one gamete that has an extra and one gamete that has not enough, and two gametes are just fine. But regardless, if these are egg and sperm, when they come together, then you're going to either end up with 45 chromosomes, meaning that you are missing one, or 46 is the normal number, or 47 chromosomes, meaning you have an extra one. And that's where things like Down syndrome come in. You end up having one of those gametes that had an extra chromosome come together with another gamete, and you have an extra. Here's another example of an instance where maybe that happened twice. So this person has 47, actually more than that, 46, 47, 49 chromosomes. They actually have 4 X's and a Y. So some kind of craziness happened and they joined together. If you have extra X's or extra Y's, you have what's called Klinefelter syndrome. You can survive. Most of these X's would then be inactivated. Here's another example. This is called Turner syndrome. So they are actually missing the Y chromosome. They do not have one. You can see an X but no Y. So they have 45 instead of 46, and it's denoted as an X and an O. Another thing kind of just randomly tacked on here at the end is the idea of mutations, and I think that you can get a basic understanding of what that means. If a chunk of a chromosome is missing or a chunk of the chromosome gets duplicated, then you start again messing with dosage issues. You either have not enough of particular genes or too much of particular genes, and then that can yield some health side effects. If it's large enough, you might be able to see it on a karyotype, but if it's just a singular gene, then you might not be able to see it on a karyotype. You can also have inversion, which means part of it gets kind of flipped around. You can also have something called translocation, where little chunks of one chromosome migrate or swap places with chunks from another chromosome. Okay. The last couple terms I want to talk about have to do with ploidy. And this ties back to the chromosomal disorders of having an extra or not having enough. Aneuploidy is just a word for not the right number of chromosomes. So we have haploid for half, which our sex cells have, diploid for 2 sets, which is what we normally have in a fertilized zygote or our normal body cells. But aneuploid means you have the wrong number. So monosomy means that you are missing one. Trisomy, like trisomy 21 in Down syndrome, means that you have an extra one. Polyploidy is a little bit different. Polyploidy, poly means many. So you actually have an extra set. So instead of having 2N and being diploid, you might have 3N or 4N. And that's a huge gene dosage problem because you have a whole extra set or 2, which means that it's really rare in mammals or really animals altogether, but it happens a lot in plants. These last couple of slides kind of are the same exact slides from earlier. I just forgot to delete them, I guess. But it kind of rehashes the idea of genetic recombination and what that means, and if they look like their parent or their offspring, and what a linked gene is, and so on, and how you actually generate a linkage map. So we kind of already covered that, so I'm going to skip it here.